Morning everyone. I'm Mgata Gautam Sudan from Central University of Tamil Nadu. Today I'm going to explain marine microbial community, particularly bacteria. Let's decode the topic. So first, what is marine? So marine is a term which is used to describe the thing which is related to ocean or sea, and it could be a plant or animal or anything which is related to the sea or ocean. And what is microbial community? So microbial community are the group of the microorganisms that live together in a shared environment and interact with each other. So as you can see, it is very interesting to observe and know what is under the ocean and so the organisms. And I feel very interesting to start this topic, and I hope you'll also feel that by this end of this presentation. As we proceed further, these are the contents which we deal with in this presentation: the introduction and the bacterial structure and how the bacteria survives in the ocean and the different types of bacteria, their ecological roles, ongoing research. So the overview is like the microorganisms which are present in the ocean or sea are considered as marine microorganisms, and they are diverse because they include bacteria, archaea, virus, protozoa, fungi, and a lot of animals are also involved in this. And the importance is like these make up 70 to 90% of the biomass in the ocean, and they're also crucial for the nutrient cycling, most of the ocean photosynthesis, and for the global oxygen production. So these are the major contributors. And marine microbiome is a collective term which is used to refer these marine microorganisms, their lifestyle, and their ecological roles. So coming into the structure, so here you can see the cell wall, cell membrane, and the usual 70S ribosome make a DNA, and the motility structure, flagella. So you can ask what is the difference from this bacteria in the ocean to the bacteria in the terrestrial ecosystem. So we will discuss this in the further topics. So as they will undergo certain structural changes in the cell membrane and the compositional changes in the cell wall, and there is a role of these ribosomes and protein in their adaptation to this extreme environment. So this will be discussed in the further slides. Can we survive in the ocean? What do you say? Let's see. So these are the expectations when we go to the oceans. We think we can survive with oxygen cylinder. Definitely Chandrasekhar will say, yeah, we can survive the oxygen cylinder. So but let's see the reality. So this is the reality after the oxygen cylinder is over. This is the reality. We can't survive for a long period of time in ocean. But how do you think the bacteria can survive in the ocean? So there are various strategies for the survival of the bacteria in the oceans. First is the nutrient acquisition. So first thing is the photosynthesis. So photosynthesis, when you talk about so two organisms, so we need to discuss. So first is the cyanobacteria. This is an abundant organism performing oxygenic photosynthesis, and it uses chlorophyll A to perform the photosynthesis. And another is a purple sulfur bacteria, which uses sulfate as an electron donor in an anoxygenic photosynthesis. And next is the chemosynthesis. So chemosynthesis is done by the chemoautotrophic bacteria. They obtain energy from the oxidation of the chemicals, which could be organic or inorganic, and this chemosynthesis is abundant in the region so where the sunlight is absent. And organic matter utilization are the heterotrophs, so they break down this organic matter from the dead remains and restore the vital nutrients. So these heterotrophs are essential and crucial in the marine environment as they restore the vital nutrients. So for them, the carbon and energy source both is the organic matter. So this decomposition process done by these heterotrophs is vital for the nutrient cycling and maintaining the higher trophic levels and also the food web. And next, getting into the adaptation to this extreme condition. The first adaptation we can discuss is the osmoregulation. So this bacteria segregate osmolysins such as betaine and trihalose, so which will help them balance the osmotic pressure which is being built up, and they can also modify their proteins such as they accumulate the more acidic amino acids at the surface, so which will resist the denaturation from the salt-induced stress. And coming to the temperature tolerance, so as you know, the cycrophiles and the cycrotrophs, see, these contain flexible membranes and produce antifreeze proteins, which prevent the ice crystal formation within the cells. And thermophiles and hyperthermophiles, which survive in the extreme hot temperatures, have special proteins which increase the stability due to the increase in the number of ionic bonds and hydrophobic bonds, and which will help in the functioning of these bacteria in such extreme conditions. And the other adaptation is like the membrane lipid changes can be utilized by these bacteria, which help them survive in such extreme temperatures also. And light protection, so certain bacteria produce pigments such as carotenoids that absorb harmful substances such as UV light majorly. An example is a Vibrio, which secrete protoxin, so it also has antimicrobial properties and absorb UV light, which is essential. And also biofilm formation acts as physical barrier, providing protection against the UV light. And we can say that the community living enhances the survival, so which implies a proverb called unity is strength. So here also the unity strength is applicable in the ocean. And coming to the survival strategies in extreme condition, so first one we'll discuss the halophiles. So halophiles, uh, there are various halophiles such as light, moderate, and extreme halophiles depending upon the salt concentration. So they accumulate the compatible solutes like potassium ions and also the oxygenic and osmolytes such as glycine butane, which prevents the water loss and protect the cellular components. And also they modify their proteins and accumulate lot of negative charge onto the surface, so which will resist this high salt concentration, and also produce bacterial rhodopsin, which is not only involved in the nutrient assimilation but also absorb the UV light. And next, getting into the pressure resistance. So these bacteria attain the structural resilience by increasing the unsaturated fatty acids in the membrane, and also they secrete certain enzymes so which enhance the stability and activity of the bacteria and maintain the functional conformation. And certain bacteria also switch the metabolic pathways depending upon the availability of the nutrients and energy in this nutrient poor environments. And next, dealing with the community interaction. These community interactions can be positive and negative, but majorly the predation and competition has a major impact on this marine bacteria. So firstly, we'll discuss the bacterial phases. So these are the major predators of the marine bacteria, and the next are the protists, which graze and alter the bacterial community and the composition and diversity. Majorly, the competition for the limited resources like nutrient and space leads to the release of the antimicrobial compounds, and also it is a boon for the development of the resistance. And coming to the horizontal gene transfer, this predation and competition, as discussed, will favor the conditions of the horizontal gene transfer between the bacteria, and it can occur through transduction, the virus, or conjugation, that is cell to cell contact transfer, or it can occur to the plasmids also. So it's prominent in the bacteria which are in close proximity, usually in the biofilms. And also the genes which are usually transferred are the antibiotic resistant genes, nutrient acquisition, and stress tolerance. So this promotes the genome plasticity and the evolutions in the marine bacteria. And getting into the ecological roles. So first is a nutrient cycling. So here you can see the carbon cycle, the carbon dioxide coming from the atmosphere into the ocean, which is fixed by the phytoplankton. This phytoplankton will be grazed by the zooplankton, so which will respire and add on carbon to the ocean, and also it will excrete, so which is an organic matter. So that implies it's also carbon source. So this dead organic matter also fluxes into the deep ocean, and when this phytoplankton and zooplanktons undergo senescence, so when they are dead, so their dead remains also sinks into the deep ocean. So here in the deep ocean, so this dead remains of this zooplankton phytoplankton can be consumed by the other organism which also respire and excrete and add on carbon to the ocean. And so this dead remains are decomposed by the bacteria and increase the dissolved organic carbon, so which will be physically mixed throughout the ocean. In this way, the carbon is fluxed in the ocean. And observing the oxygen cycle, as you can see, the photolysis is splitting of the water into the oxygen, and also the weathering process, that is the breakdown of the oxygen containing minerals from the rocks which will give out the oxygen. And also the photosynthesis increases the oxygen in the environment, and also the respiration decay, and also mainly in the marine sediments, so the decaying of the dead remains while degrading the organic carbon, so there is a release of oxygen into the environment. And observing the hydrogen cycle, so it is simple but it majorly circulates through different states such as water, molecular hydrogen, methane, hydrogen sulfide, ammonia. So but it is linked to the carbon cycle and oxygen cycle. So this glucose is converted to hydrogen, and hydrogen is converted to water and further converted to molecular hydrogen. So in this way the cycle is repeated, and this hydrogen cycle is connected to carbon cycle and oxygen cycle. So observing the nitrogen cycle, here you can see the nitrogen enters from the atmosphere into the ocean, and there it is fixed by a bacteria into nitrate, and this nitrate is used by the phytoplankton, so which exceeds the ammonia, and this ammonia is synced or mixed into the benthic zone, and so which is again nitrified by some other bacteria into nitrate. So this nitrate is vertically mixed throughout the ocean, and also this nitrate is converted back to the molecular nitrogen by the denitrifying bacteria. So observing the phosphorus cycle, so this phosphorus cycle is mostly from the weathering and erosion process, and the phosphorus enters into the ocean, and the dissolved phosphates are consumed by the marine plants, and marine plants are grazed by the marine animals. So when these are dead, so they create the sediments at the ocean bed, and this will create sedimentary phosphate rocks. And observing the sulfate cycle, so sulfate also enters the ocean through majorly through river, atmosphere, and the sulfate is the most abundant form which is present in the ocean, and sulfur oxidizing bacteria convert this sulfate to sulfide, and at the ocean depth the sulfide is converted back to the sulfate by the sulfur oxidizing bacteria. And coming to the iron, so iron is required by the phytoplanktons for performing the photosynthesis. So in this way, it is involved in the conduction of many biogeochemical cycles, and also it is involved in the primary production. That is, it can perform the chemosynthesis and also photosynthesis, and this is fundamental for sustaining the marine food web. And also it is essential for the decomposition. As you can see, the phytoplanktons, uh, they are being raised by the zooplankton, and after the death and their excretion contributes to the organic matter which is decomposed by the bacteria, and this restores the nutrients, and the marine microbes are responsible for producing more than half of the world's oxygen through photosynthesis. And also these have a influence on the greenhouse gas dynamics, and also these regulate the earth's climate, because so they control this carbon cycle, oxygen, nitrogen and sulfur cycle. And this bacteria also establishes symbiotic relationship with the coral reefs, which will be further discussed in the slides. And coming to the pathogenicity, they can become pathogenic, and that's why we have to study and monitor this microbial community that will be an indicator of the environmental health. And getting into the biotechnology potential, so here you can observe these marine microbes produce enzymes, fungicides that are used to produce toxin, antibiotics, antifouling agents, and they produce biosensors, and also marine organisms, as discussed, they produce UV protective compounds and a lot of compounds which are of biotechnological significance. So these marine bacteria are a promising source of novel bioactive compounds with application in medicine and industries. So as these marine bacteria are for performing these many ecological roles, in my point of view, marine bacteria are the true heroes. So they also deserve some elevation. So it's time for the elevation of marine bacteria. So I hope you enjoyed the elevation. And coming into the types of the bacteria. So these types of the bacteria in the ocean depends on a different criteria. And here the first criteria we're considering is energy source. And firstly, is the phototrophs. So they use light energy for fixing carbon, as we know. So other than cyanobacteria, the examples are cyanococcus and prochlorococcus. And next, coming to the chemotrophs. So chemotrophs, as you know, they utilize energy from the oxidation of the chemical compounds, and examples of these include nitrifying bacteria such as Nitrosomonas, Nitrobacter, or sulfur oxidizing bacteria such as Thiobacillus. And next are the heterotrophs, so which are essential as the decomposers, for example, Vibrio, Pseudomonas, and also pathogens such as Streptococcus. And based on the habitat, first comes the planktonic bacteria, so which are present at the epipelagic zone. And here, what you'll observe is the pelagic bacteria, which is abundant bacteria in the ocean. And next comes the benthic bacteria, so these are almost present at the ocean floor, and methanogens are the predominant members. The cyclophilic bacteria, which survive in the cold environment, that is polarinonas, and thermophilic bacteria we survive in the hot environments, and the example is a thermotoga. Based on the function, so there are nitrogen fixing bacteria such as rhizobium, and also sulfur cycling bacteria such as the sulfovibrio, and carbon cycling bacteria such as pelagibacter and Shonella. Next, getting into the final segment of the presentation, that is the recent research and advancement regarding the marine bacteria. So first is the discovery of this Thiomargarita magnifica. So this is the largest bacteria, so which was measured 1 centimeter long and was discovered in the mangroves of Gandolip. And next is the oceanic bacteria form transspherical communities. So these form spherical communities in order to take different roles to break down the large food sources more efficiently, so that they can degrade efficiently, so that they are forming the spherical communities. And next is the Poriferispera heterotrophicus. These are the novel species in the ocean seabeds, and are known for their unique budding mechanism. And coming to the arsenic breathing life discovered in the tropical Pacific Ocean. So here the bacteria are able to survive by breathing arsenic in the oxygen poor parts of the ocean. And next, the genetic modification of the marine bacteria. Here, the metagenomic data reveals that there are intra-species population structure which are present in this marine bacteria. And also, you can observe that these bacteria break down the medicinal compound, further nitrogen requirement, and also as discussed earlier, so this bacteria gets associated with the coral reefs, so which are being threatened. So these are the recent research and advancement regarding the marine bacteria. I hope you have gained a lot of information regarding the marine bacteria. Thank you, Kavya ma'am, for giving me this opportunity. And also thank you, my dear friends, so for listening to my presentation patiently. So this is Gautam Sudan signing off. Thank you. Wait. So after explaining all these and after me explaining you and describing these marine bacteria, I think I have to give a tribute to this marine bacteria. So I would like to conclude my presentation with a small tribute to the marine bacteria. All hail the bacteria, all hail the marine bacteria. Thank you.